So today I'm going to be talking about AquAdvantage Salmon, which is a genetic modified salmon that grew faster than ordinary salmon. So today I'm going to be talking about AquAdvantage Salmon, which is a genetic ultra salmon that grows faster than ordinary salmon. So a genetic modified organism is basically an organism whose genetic material, which is DNA, has been altered in a way that does not occur naturally by mating or natural recombination. By which I mean is us humans altered their DNA to manipulate the organism's genome using biotechnology in order to produce improved organisms that have our desirable traits. An AquAdvantage salmon is a genetically modified Atlantic salmon developed by AquaBounty Technologies, whose gene is altered and the speed of growth is increased, but the quality of the fish is not affected. So, the process of creating an AquAdvantage salmon is to insert the growth hormone protein coding sequence from a Chinook salmon and a terminator and a promoter from the ocean-path antifreeze gene into an Atlantic salmon. So antifreeze protein in a fish's blood gives the fish the ability to survive in the near-freezing waters. And the Chinook salmon's growth hormone coding sequence leads to a higher concentration of the growth hormone in the blood, causing the genetically modified salmon to grow much more rapidly than it would naturally. So the pictures below shows that in order to insert the desirable gene into the Atlantic salmon, we need to first insert it into a plasmid. So the plasmid then can replicate into billions of copies into a bacteria. The growth hormone separates from the bacteria, creating a linear bed of DNA. Afterwards, the cultivated DNA mixes with fertilized Atlantic Ocean salmon egg, so that when the eggs hatch, some of the fry are genetically altered, some are not. This picture illustrates the whole process of creating AquAdvantage salmon. So the advantages of genetic modified salmon is that the new inserted gene produces a hormone all year round instead of only in summer and spring. And the ordinary salmon need most 7 years to reach reproductive age, while an AquAdvantage salmon takes 3 years or less. And with year-round growth hormone, a genetic altered fish reaches full size in less than half the time, which is 16 to 28 months, instead for 36 months for an Atlantic salmon. The maximum size of an ordinary salmon is 13 inches, in comparison to an AquAdvantage salmon, which is 24 inches. The pros of creating AquAdvantage salmon is that a genetic modified salmon eats 25% less food than ordinary salmon, so we spend less money to create salmon that grows faster and grow larger. And it reduces pressure on wild fish stocks, and it could help meet rising demand of fish. And the cons are scientists on long-term effects on genetic altered salmon is limited. And the food safety activist and fishermen say genetic altered salmon won't be properly labeled. This graph shows the weight of AquAdvantage salmon compared to standard salmon. So in terms of growth, the salmon have wide variability of phenotypes that allows them to adapt to variety of environment conditions. However, even genetically similar fish may have very different phenotypes when exposed to different environments. So, transgenic fish fed to satiation in hatchery conditions grew almost 3 times longer than controls, while transgenic fish in a simulated natural environment grew to be only 20% longer than controls. This shows that the environment affects the size of transgenic fish. And these pictures indicate like the growth hormone fish and wild-type fish body size differences. Many studies have found that overexpression of growth hormone can result in changes in a variety of traits, including behavior, swimming abilities, and body structure in salmon. And the body malformations found in salmon overexpression growth hormone can mean the fish might not be able to swim as fast as wild-type fish, but the FDA's 7 years data indicating no difference in animal health and welfare between AquAdvantage and wild-type salmon. So now I'm going to be talking about the process of inserting the DNA into the plasmid. So a plasmid contains three elements: cloning site, replication origin, and a drug-resistant gene. So the cloning site is where the foreign DNA fragments can be inserted, and the drug-resistant gene destroys antibiotics to allow selective growth of the host cell. And the replication origin is to allow the plasmid to replicate in the host cell. So a restriction enzyme, which is the EcoR1, cleaved the palindromic sequence GAATC in the cloning site to create single-stranded sites, sticky ends. And the foreign DNA is also digested with EcoR1, and then the sticky ends of the foreign and plasmid DNA hybridize and are sealed into phosphodiester linkage by the enzyme DNA linkase to eventually create a recombinant plasmid. And we add the E. coli bacteria to the recombinant plasmid through a process called transformation. A few cells take up a recombinant plasmid while most other cells do not. So we usually pour the bacteria into the plate contains antibiotic and penicillin, but in the process of creating like an AquAdvantage salmon, we only use bacteria to replicate the gene and form linear bit of DNA. We can then add the DNA into fertilized fish eggs.